Hello, dear friends. Today I am going to discuss DNA damage with you. I am Dr. Sujata Magdum, Assistant Professor in Zoology from KTHM College, Nashik. So, as you all know, DNA is a very important biomolecule of life because it is concerned with transmission of characters from generation to generation as it contains the information stored within itself. Apart from that, it controls all the bodily activities for the normal life. Now, to begin with, we will first understand the basics of DNA. You all know that DNA cannot be completely broken down to monomers and rebuilt into an entirely new one from the scratches. This is because it requires a pre-existing single strand of DNA which act as a template. Moreover, a free 3' OH group is needed for the synthesis to begin with. DNA acts as a single blueprint that contains all the information to make a new copy of any other polymer molecule in the cell, and therefore we can say that the genetic information coded in the DNA should be very accurate for normal functioning of the cell. Now, we all are familiar with the fact that preservation of the genetic information in its original state depends on the accuracy of its copying during DNA replication. Moreover, timely repair of any event which may compromise the integrity of the DNA molecule or may change its building block or their arrangement in any manner can lead to DNA damage. Now, what is the need to study the DNA damage? This is because DNA, as I said earlier, is concerned with preservation of genetic information, transfer of progeny in correct form, and is essential processes like transcriptions as well as translation of DNA leading to synthesis of protein totally depend on the integrity of DNA. If at all there is any kind of damage seen into this, it can lead to gene expression, and further this gene expression which can be lethal for the survival. Now let us see what is the basic difference between the DNA damage and the DNA mutation. DNA damage is a physical entity wherein the strand of DNA can be broken down, and when this occurs, it can be identified by certain enzymes which are present into the nucleus. Further, because the enzymes are able to identify such DNA damages, it can be corrected, and therefore such damages are prevented in the nature itself. Whereas when we talk about DNA mutation, there could be base pair changes, and these base pair changes are so minute that the enzymes cannot identify them, and hence mutations are irreparable. There are various factors which are concerned with bringing about the process of DNA damage and can be categorized as endogenous factors as well as exogenous factors. Endogenous factors could be certain carcinogens or it could be certain chemical agents, whereas exogenous factors could be certain chemicals like alkylating agent, UV radiations, X-rays, and all these can lead to replication errors. As a result of which, there can be abasic sites into the DNA or there could be breaking into the DNA strand. Now coming to the breakage into the DNA strand, which are commonly called as the lesions or the damages, it can be seen into the single strand or it could be seen into both the strands as such. When these breakages are observed, these are seen in the interruptions in sugar phosphate backgrounds of the DNA, and it could be because of the reactive oxygen species or the reactions of the certain enzymes like nucleases, or there could be some changes into the base pair damages. In the same way, lesions can also be seen with reference to the base pairing, and hence These base pairing can lead to interruption in N-glycosidic linkages, which enables them to bind with the phosphate backbone into the DNA. And these interruptions are always spontaneous. Such interruptions are called as depurination, or in common terms it can be called as a base pair damage. Now these DNA damages can lead to cell cycle arrest. When such cell cycle arrests are observed into the cell, because of which the DNA replication is postponed, and this postponement is retained until and unless it is repaired. Once the DNA is repaired, then the normal functioning of the cell follows. Sometimes DNA damage can lead to apoptosis, that means damaged cells are removed, wherein we need to remove the damaged cells. B in the sense the body itself removes the damaged cells. Some of the times it is observed that all the machinery into the body mechanism it fails, and therefore such DNA damage they cannot be removed, and therefore such damages leads to genomic instability. These genomic instability are the concern factors which brings about mutation, cancers or formation of deadly diseases into the organism. There are different DNA repair systems, and these different DNA repair systems could be error-free repairing or error-prone repairing. Error-free repairing leads to complete repair of DNA damage, whereas error-prone repairing can lead to mutations. In the next slide, we are going to study in detail the different mechanism which causes DNA damage. Thank you.